Good morning, and thank you for the warm introduction. It's a pleasure to be here. This is a pretty daunting topic. I could keep you here for the next 5 years, telling you all about the new and emerging therapies in myeloma, but we'll see what we can do. I'm planning to talk for less than the allotted time, so there's plenty of time for questions and discussion at the end. In terms of disclosures, in my work as a researcher with the Canadian Cancer Trials Group, I have received research funding from many pharmaceutical companies, including Amgen, Celgene, Gilead, Kite, Janssen, Novartis, Roche, and others. I have no personal financial interest in any of these companies, but have worked collaboratively with many to conduct clinical research. Which way? That way. Yep. So, the objectives I thought we could lay out over the next 40 minutes or so to highlight just a few of the many emerging therapies in myeloma, to give you some understanding of why there's excitement and what particular directions the field is moving. But also, and perhaps more importantly, to give you some insight into the process and the procedure for how new drugs and new therapies emerge. Because then you can take what you learn and you can extrapolate it and use it for any new drug, seeing where it is in terms of its testing to see how much confidence you can have that that may yet be the next new big thing. And there's a lot of reason for hope, and I very much hope that that will come across during this talk, but there is also reason for caution. And you can't always fully believe every headline on the news that says this is the next blockbuster, this is the cure for myeloma. Sometimes it may be true, but sometimes it's worth looking beneath the surface just to check, does the evidence really support that statement at this point? The cast. This will make sense, but these are all characters who have contributed to my learning, contributed to this talk, and who you will be meeting over the next 40 minutes. The fireman on the left is representative of my patients, from whom I have learned a tremendous amount. You're actually a really daunting audience to talk to because this matters so much. And you may not realize how much you matter to us, but you do. On the right, my colleagues, Sita Bella being one of them, who did a fantastic Journal Club presentation on myeloma on Friday, and some of whom ideas I've used here, as well as other colleagues, including David Lee, who's given me one of his slides, and Vincent Rajkumar, who's not a colleague at all, but who has a fantastic Twitter feed. And if you want to keep up to date with the myeloma clinical research, following Vincent Rajkumar will keep you right up to speed. You'll also meet a mermaid and a manatee. So firstly, how new drugs are developed. It pretty much always starts in a laboratory, basic science. And within pharmaceutical companies, for example, there are many, many, many medicinal chemists who may be going through screens of new molecules to see, would one work in a certain disease? Or perhaps coming at it from another direction. If you know in a given disease there is a particular abnormality that maybe you can target with a new type of medication, then they may be trying specifically to engineer and design a chemical compound that will attack that vulnerability in the cancer cell. Sometimes it may also be going out to the Amazon rainforest or somewhere else, or discovering plant products that are already there and determining if indeed they do have medicinal properties. Once in the lab, you identify a product that you think may have potential. It goes from the in vitro Petri dish test tube testing into small animals. And these are ones where you think you have some idea it might work, but you need to check to be sure before you would take it to a human. And most commonly, these are done in mice, and the animals are very well cared for. There are extremely strict guidelines which must be followed and are rigorously checked for to ensure that animal welfare is protected. But before a drug ever gets into a human, it has been tested, or I should say a potential drug, it has been tested in at least 2 different types of animals, because clearly we are different than animals. And just because something looks really, really, really good in a mouse, it may not work in a human. And there are certainly examples of that. Another relevant finding is that side effects very much differ between species. So many of you will know chemotherapy and cancer treatments can cause nausea and vomiting, and commonly do. Now, we don't test these drugs in horses, but if we wanted to, it would be no good, because who knew horses couldn't vomit? And it's why, if any of you are vets, apparently it is very, very, very serious if a horse gets trapped wind or needs to vomit because they simply can't. So then we move on to a first-in-human study, and these are always a big deal. Where you are testing, is this drug really safe? So anyone conducting and anyone enrolling in a first-in-human study will be really carefully monitored to see, is this safe to give? What sort of side effects does it cause? And you don't often, being completely honest, you don't often expect to see benefit in a first-in-human study, because you are going in so cautiously, usually, at a low dose to understand more about the drug. So that would be your first, is starting off at a low dose and then gradually increasing the dose in more people as you get more confidence in the drug's safety. And if you prove in a small number of people it looks safe, it looks promising, well, then you'll conduct larger clinical trials, testing in a greater number of people. Does it work as well as you hoped it might? But all of these studies in people will be complemented by studies in the lab. So blood samples may be taken, or sometimes bone marrow samples taken, to understand, is this drug working the way we thought it was going to when we designed it? Or is there a side effect that actually means this drug is going to be no good? And maybe we should re-engineer the drug to get rid of that molecule and replace it with another, so it's easier for someone to take. Or can we identify in the laboratory, based on testing, a group of people who may benefit more than others, a biomarker that would then let us know this is the drug for them, and this drug is unlikely to benefit someone else. So this loop is continuous, and depending where a drug is in this loop of testing, depends on how much confidence you can have in its likelihood to improve lives. And I know we've got a whole dedicated panel session to clinical trials next, but just a brief introduction to the different phases of clinical trials. Phase 1, is it safe? And often with that, what is the recommended dose as you work your way up through the lower doses to see where you think the best dose is going to be? So, for individuals enrolling on a phase 1 trial, while there may be benefit, that certainly cannot be guaranteed to that individual. So we are always extremely grateful to people who are willing to put themselves forward to help contribute to science in this way. And typically, phase 1 trials are for people who have already gone through most of the effective therapies and ready to try something potentially new. Phase 2 studies are once we've figured out what we think the dose should be, does it work? So you take a group of people with a known disease, you give them the drug and see if it has the benefit that you hope it's going to have. And then phase 3 is a randomized study where once you've passed phase 1, you've passed phase 2, you take a group of people and you randomly assign them so they are similar in both arms to either your new drug or the current standard of care to find out, is it better? And really, once you've got phase 3 data confirming it's better, that's where you know you're onto a winner. Everything before that is very promising and sometimes can inform and change practice. And phase 4 is a little bit separate, but then once a drug is out in the real world and once people are getting it, is it working the way you thought it was going to work? because in a clinical trial, it can be a little bit sterile. You take a very select group of people, and any of you who have participated know you have to meet different criteria. And then in the real world, often it's used in more individuals, and it may be used at other centres. So just checking, are the phase 3 results really what we thought when everybody's using it and getting it? Okay, so now actually on to the new and emerging therapies. And my point here is it doesn't always have to be that shiny and new. Sometimes building and improving on agents in a known class can be really helpful. So Bortezomib is a proteasome inhibitor. It's also known as Velcade. And we use it very, very commonly in Canada. Many of you have probably had it. We've been using it since around 2003. It's extremely effective. Over that time, we have understood that while we used to give it intravenously, we can now give it subcutaneously, which makes life a little bit easier. We've also realized that while we used to give it twice a week, now we can give it once a week. And this seems to reduce the side effects, particularly peripheral neuropathy. Now, clearly there's variation in practice, but we continue to learn even once we have an established drug. Second generation drugs are coming along now and are exciting. They're still proteasome inhibitors, but they bring something new. They act just a little bit differently. Carfilzomib is one that was relatively recently approved and relatively recently adopted into practice. It seems to be a bit stronger than carfilzomib. And sometimes in people who've previously had bortezomib and maybe it didn't work as well as hoped, carfilzomib can work. Ixazomib, another new one, and currently some people are getting it through a compassionate access program from the drug company. One of its exciting benefits is it's a tablet. So you don't need to come for the injections, which we know are a real pain when you're coming every week for indefinitely, or twice a week, even worse. So a tablet, wouldn't that be nice? And there are many others coming. Learning how to say these is always a challenge. Proteasome, I didn't say that right, or Bortezomib, and many others in development that may offer additional benefits. Time will tell. But I also want to make really clear, just because we've got shiny new ones coming, doesn't mean there's anything wrong with the old one. And actually, Bortezomib still remains an excellent drug, still remains in widespread use. We've got great experience with it, know how it works and what to expect. So the shiny and new is great as an add-on, but it doesn't replace the old. Moving on to something different, but also exciting. Monoclonal antibodies. On a cell, there are all sorts of different markings on the outside of that cell that we can use to identify in the laboratory. Oh, I wasn't expecting that. We can use in the laboratory to identify what that cell is. Now, while we give them names, such as CD20 or CD38, that's just for our convenience. They all have functions. They may be letting sodium into the cell or potassium out of the cell, but we give them names to help us recognize them and then use that to help us identify and target and design new drugs. The benefit being they may be much more specific than chemotherapy. Because chemotherapy, fantastic, but indiscriminately attacks all rapidly dividing cells. And you know that from the hair, which grows rapidly and can be affected. You know that from the gut lining, which is continuously renewed roughly every 3 days, which is why chemotherapy can be really hard on the gut, the mouth, causing diarrhea and ulcers. When you can target a monoclonal antibody, it's not perfect, but you can target a specific type of cell. And the disease in which we have the most experience so far with monoclonal antibodies is probably lymphoma, because B lymphocytes have what we call CD20 on the surface. And there's a drug called Rituximab that we've been using for well over a decade, probably close to 2, that targets CD20 and eradicates the B lymphocytes. Now that's great when the B lymphocytes are your lymphoma cells. Now the astute of you will also be noticing that means it's also affecting your normal B lymphocytes, so it's not perfect, but it's a lot better. So that's the principle of monoclonal antibodies. You design a drug to target a marker on the surface of the cell. And daratumumab, I know Dr. Rees talked about this yesterday. It's pretty exciting in myeloma. We've been watching this one coming for a few years, and we're delighted to see it's arrived. So it has been through the preclinical, the phase 1, the phase 2, the phase 3. We know it works. Oh, I seem to have left a timer on accidentally here. What daratumumab is targeting is CD38. And CD38 is the marker on the cell surface of plasma cells. Plasma cells being the malignant cells in myeloma. So daratumumab binds directly to the plasma cells, leading to cell destruction, which is exactly what you want to happen. Now, it also does bind to others. So on the left is what we want to happen. The daratumumab, CD38, binding to the plasma cell and leading to its destruction. Of course, it does also damage the normal plasma cells, but not so much the other tissue. And the red cells, the red cells is a really interesting one because CD38, it's not solely expressed on plasma cells, and there also is a little bit on the red cells. This doesn't tend to cause major problems, but it does mean if someone on daratumumab ever needs a blood transfusion, it can be really hard in the blood transfusion laboratory to get the right blood type for them and to match it because it interferes with the cross-match testing that always happens. So, having learned this through the clinical trials, now before anyone starts daratumumab therapy, there's an extended phenotype-genotype panel to be sure we know in great detail what an individual's blood group is, so that if they then need a blood transfusion while on treatment, it can be more safely administered. The other issue with monoclonal antibodies can be infusion reactions. And that has been an issue with this drug, which has got easier as we've learned more and realized that it's really important to give prophylactic preventative medication to try and prevent these infusion reactions, and that certainly has resulted in a reduction. The other issue, which is a real pain in your life, and also a challenge in a hospital when you're trying to get everybody treated in a set time, is it can take a really long time to infuse, up to 8 hours over 2 days for the first dose, for example. So there's a very exciting study that's being done by the Myeloma Canada Research Network, which is looking at trying to safely administer daratumumab in a much shorter time. And while it's still early, so far, based on the initial patients who've enrolled in this study, it looks really promising. So that would be very nice if we could give this drug, but in a shorter time without many of the serious infusion reactions. And down the line, what will be even more convenient, there is a subcutaneous formulation in development that still needs more testing, but may also help make life easier for people. And this is a personal story. This is one of my patients. From 2 or 3 years ago, fantastic young guy, firefighter, who had absolutely horrible myeloma. And I learned a tremendous amount from him. Sadly, his disease was really aggressive, and he burned through multiple lines of therapy with not great response at all. He enrolled in a clinical trial, PHL 091 out of Toronto, that did help some people. It didn't help him. And then this was happening at the time when we were just starting to think daratumumab was going to be a big deal. But it was not yet available in Canada. And this isn't about me, but I actually got really upset about this case and did a lot of effort to try and secure the drug for him through insurance companies, through Health Canada, through the drug company, and I didn't manage at first. But he was absolutely fantastic. And while I certainly tried not to project my anxiety to him, he clearly was perceptive. And he talked to me during one consultation and said, "It's okay. I've benefited from everything, all the developments that have occurred up to now, and I'm very grateful for that. I've tried to contribute to the next developments through participation in a clinical trial. Maybe that will help people that come after me." And he said, "I know there will be more advances in the future, but I've had my chance. It's good enough." And I thought that took tremendous poise and grace. We actually did end up getting daratumumab because then the drug company did open up a special access program and he got it. It reduced his myeloma numbers. I'm not sure it really made much difference to his overall life expectancy. But I learned a lot from him. That does not mean I will not continue to advocate, but also a realistic approach, being grateful for what you can benefit from already, while still hoping for more and always continuing to hope for better. Oh, there's one more little story. His wife. So after he died, his wife and I had a great chat, and she brought to me a beautiful little gift. It's just a recycled glass jar with a painted lid and a little chalk marker on the edge. And it's for every year. My daughter Daisy's here, she knows about this one. Every year, over the course of the year, you put in just a little note with your memories. And then on New Year's Eve, you open them up and reflect on the year. And she put in the first memory, which did bring a tear to my eye the following New Year. But that's a tradition that we've kept going. So while daratumumab is the first drug in class, there are more coming. Rituximab is just one more example of another CD38 monoclonal antibody. And the phase 1 study, remembering phase 1 is early, and the primary endpoint of this study was trying to get the right dose. Around 50% of people had infusion reactions, but for the most part, they weren't too serious. And around a quarter of people treated at the higher doses, their disease responded, which is all very promising. And even better, the response lasted for a little bit, about 6 months. Now, of course, you want better than that, but more studies will be ongoing with this particular drug to see if we bring it forward in earlier lines of testing, when we compare it to others, will it offer any benefits over the CD38 monoclonal antibody we now have. So monoclonal antibodies, we know they're great. We've been using them in other diseases for a long time. We're excited to now get to start using them in myeloma, and I think there'll be more to come. Right. This is a mermaid. Anyone know what the other guy is? Thank you very much. A manatee. So mermaids. Mermaids were seen by sailors for a long, long time. And it's now become apparent they may not have been mermaids, much as they believed they were. It's kind of disappointing, actually, as you read the history. They were so convinced they'd seen mermaids. They even brought a mermaid skeleton back to Portsmouth in England to be tested, saying, "Look, it's real, we've seen it." But what it turns out is that all these apparent mermaids were probably manatees or their cousins, also known as sea cows. And there are reasons why people may have been confused. Apparently, these manatees can move their heads from side to side. Their front flippers do have 5 fingers that look human-like. Their face has some human-type features. I also wonder if the rum quota that they had daily as sailors may have contributed. There's also speculation that scurvy, vitamin C deficiency, which sadly was very, very common among sailors before it was discovered, just a dash of lime or lemon will fix it. There's a suggestion that could affect vision, cataracts, or maybe bleeding in the eye. So maybe there are various reasons why they thought they were mermaids. My point is, sometimes you don't know if something that you think is a mermaid may turn out to be a manatee, which is why we always need to look really closely. Venetoclax. I'm going to take you back a tiny bit to basic science. As much as I understand, I was studying last night. So BCL2 inhibits cell death. Now that's and we all have BCL2. That's a really good thing if you want your normal cells to stay alive and stay replicating and keep you alive. But some cancer cells, including myeloma, overexpress BCL2. Now that is not a good thing. If your cancer cell is sending out a signal that says, I want to live, and managing. So venetoclax, it's a really cool drug. It is a tablet that inhibits BCL2, and in some diseases, such as chronic lymphocytic leukemia, it's been fantastic. And it's now a drug that's moving into practice and is benefiting people with chronic lymphocytic leukemia. It looks promising in lots of other diseases, and it's being tested in them. And there was a period where we did wonder if it should just be sprinkled on breakfast cereal, it looked that good. And these are the early studies in myeloma. This is from PubMed, which is where if you type in your search, you can get all sorts of things that pop up. So Saji Kumar, a fantastic guy, did the phase 1 study along with many others and identified in the phase 1 study, so no comparison, that it looked like venetoclax was going to be very effective, especially in a particular subtype of myeloma. Now T1114, that means your chromosome 11 and your chromosome 14 have got stuck together in the myeloma cells. So further testing, in part, focused on this group, thinking, maybe this is the group that are going to benefit the most. Yes, okay. Further testing went on and continued to look promising. Anyone know what I'm getting at with this? Bellini. Bellini, this is personally happy to me. Bellini is a cocktail made with Prosecco and peach juice or peach nectar. And it originated in Venice. And I served it at my wedding. It was about the only fancy thing at my wedding. And we also honeymooned in Venice where we had Bellini. My wedding wasn't all that fancy. My husband refused to do a first dance, so we got a bouncy castle and did a first bounce instead. But true story. The Bellini was fancy. And the relevance is the Bellini randomized phase 3 trial, in which venetoclax combined with bortezomib and dexamethasone was compared directly with bortezomib and dexamethasone and a placebo. So what this means is that for the 291 people who enrolled in this study, it effectively was a computerized flip of a coin to decide which treatment they went on. And nobody knew which treatment they were on. The patients didn't, the doctors didn't. But there is one group of people who do, the statistician and the Data Safety Monitoring Committee. Because for every clinical trial, there is a group of people who are looking at the study as it is conducted to check, yeah, this is going okay, keep going. So in this study, there was a pre-planned interim analysis where you look at the data and just check it's going the way you thought. And it looked fantastic. The response rate, meaning how much your myeloma markers went down, be that your IgG or your IgA or your kappa light chains or whatever your myeloma secretes, the response rate in terms of reducing those was twice as good with the venetoclax combination. Progression-free survival, meaning how long your myeloma stays away, was twice as good. The MRD negativity rate, now that's meaning your minimal residual disease. So when you're doing really granular detailed testing to see is there any trace of myeloma left, it was 4 or 5 times better. And in terms of the serious adverse events, so really bad complications, it didn't seem to be an increase. This looked fantastic. But there is a major twist. Twice as many deaths in the people who were assigned to the venetoclax-containing arm. Now, this has caused a lot of concern, and rightly so. The FDA, that's the Food and Drug Administration in the US, who oversee all trials, they're the equivalent of Health Canada for us, have put all these trials on hold. And there is a huge amount of work ongoing at very senior and high levels to try and understand why. How can a drug look so good, but then cause twice as many deaths? Because clearly none of this matters. Well, I'm overstating that slightly. Quality of life is also hugely important. But if we have defeated the disease, but in the process defeated the host of the disease, the person, that's no good. That's not what we're looking for. So, venetoclax, I think it's yet to be determined if it's the mermaid we thought it was going to be, or if it might be a manatee. So this is one to watch this space. And it's quite interesting, in the US, they very quickly adopt new treatments. So venetoclax has actually been in use in the US off-label, so out of indication, for some time. They have all pulled back now. Many of the senior physicians have pulled back and are not sure if it should be used. So it's one of the interesting things in Canada. I know sometimes we get agitated that it takes us a little bit longer to get drugs, but sometimes there's benefit to that when you can understand more about its benefit and about its side effects before you launch right in. So BCL2 inhibitors, good for CLL. Not yet clear if it's going to improve the lives of people with myeloma. Right. BCMA. I think this is going to be important, but having told you everything I've told you, I hope you're looking back at me saying, prove it, I want to see the data. So BCMA stands for B-cell maturation antigen. Now, vaguely, like we mentioned, CD38 for plasma cells, BCMA is also on the surface of plasma cells, including myeloma cells, but it's actually a bit higher than it is on the ordinary plasma cells. And BCMA is being targeted in all sorts of new drugs that are in testing to see how can we design something to hit this BCMA target and use it to knock myeloma back even further. And there are various different approaches, including BCMA ADC, stands for antibody drug conjugate, CAR-T, chimeric antigen receptor T-cell therapy, and BiTEs. And we'll very quickly go through each of these for the broad principles. So this is early work. Early work in which Canada is leading. So this particular study that's catching international attention is led out of Toronto. Dr. Suzanne Trudel is the principal investigator leading this. It's open in a few centres across Canada, the US, and the UK. And she and the team really have been leading the way for this particular drug. So what you can see, the GSK 2857916, that tells you that's the drug's name right now. It's not that catchy. GSK tells you that it's a drug produced by GlaxoSmithKline. And the number tells you how hard they work to try and generate new compounds and how many they go through before they find one that might be a winner. If this proceeds through further testing. It will be given a name. It's possible I'm behind the times and it may have one in the pipeline. But this particular antibody drug conjugate, it's like you're taking your monoclonal antibody, so you can directly go and attach to your myeloma plasma cell, but you're attaching to that monoclonal antibody another treatment, so that when it binds, the BCMA monoclonal antibody piece can go right inside the cell, the cancer cell, and then it can release the chemotherapy or the targeted drug or whatever it is right inside the cell, with the hope of making it much more effective and the possibility of perhaps reducing side effects from other cells. Now, in this phase 1 study, eye problems were a bit of an issue, actually, particularly the cornea, and I know the team have been working hard to try and figure out how those can be mitigated, and low platelets that can predispose to bleeding. But we know almost all of the drugs we use cause low platelets, so that's one we're quite familiar with managing. And in this phase 1, 60%, so around 6 out of every 10 people, had some benefit. So that certainly shows promise. And the median duration of response, so how long they got benefit, was for around a year, clearly with a wide range. And anytime someone shows you a median, think about the range. There will be some poor folk who got nothing from this drug. And if the median's 1 year, that means there are some who are continuing to benefit out beyond that. So it's promising, but we need to know more. We need to know more than a phase 1 to determine if it's safe and going to be better than the others. This is full-on science. BiTEs. We're still talking about BCMA, but this time we're looking at bispecific T-cell engager monoclonal antibodies. So stick with the same principle. You've got your monoclonal antibody that's catching on to the myeloma cancer cell and sticking tight. But this time, to that drug, you add another anti-CD3. Now, CD3 attracts T lymphocytes. And this is where it starts to get cool. This is where we are finally, the greater royal we, are finally starting to figure out how we may be able to harness the immune system to actually kill cancer like it should. So when you bring together the T lymphocyte, which is an immune cell, in direct proximity, so it's physically attached to the myeloma plasma cancer cell, the T lymphocyte, the body's own immune system, destroys it. And even better than that, because the role of the immune system is to learn to recognize foreign things and destroy them and augment that response, the T lymphocyte then replicates, proliferates to make many more that can go and attack other plasma cells. So again, this is really, really early, and so far it's been a handful of individuals treated, but the results look like they might be really, really promising. So this is another one to watch this space. Now the challenges are going to be, it has to be given as a continuous infusion because of the way the drug is made. And so that would mean, in practical terms, the way it is right now, walking around carrying a pump, continuously infusing this drug, maybe having to change the pump it depends and it's under discussion, maybe every 48, 72 hours. So there is some inconvenience there. It's also quite a difficult drug to manufacture from what I understand. So there likely would be cost implications for that in terms of what the end product may cost, but definitely worth pursuing. And then chimeric antigen receptor T cells, or CAR T cells. Now there is a huge amount of excitement and hype and hope around these. I hope it comes true, but it's again still very early days. Emily Whitehead has become the poster child for CAR T cell therapy. She's an American kid who unfortunately got acute lymphoblastic leukemia around age 6. Around 90% of individuals with acute lymphoblastic leukemia in children can be cured. But her disease, for some reason, was particularly aggressive and resistant to all of the usual treatments. She'd had chemotherapy, she'd tried different chemotherapies, and it just kept coming back. She was the first child treated at the University of Pennsylvania with CAR T-cell therapy. This is her on the right. It's probably a couple of years ago now. Apparently cured, around 7 years out, walking her dog called Lucy, playing soccer back at school, living life to the full, and actually a really strong advocate for expanding and learning more about CAR T-cell therapy. She actually went to the FDA as part of the initial meeting to try to get the drug approved for ALL, which was successful. And Carl June is an incredible man, one of life's geniuses, I think, who really pioneered this. He designed the treatment she was given, oversaw her treatment, including when she became critically unwell, because these drugs can have really serious side effects, including when she became critically unwell, going back to the laboratory with her blood to figure out what's causing this massive inflammation, and think really laterally to then take that information, find a drug used for rheumatoid arthritis, and give it to her, leading to her very rapid recovery and survival. So Carl June was Time's 100 most influential people of the year listings last year, and actually Emily wrote the summary proposing him for it. So it's a really nice story. Now, how CAR T-cell therapy works, it's actually, in the initial phases, not that dissimilar to an autologous stem cell transplant. So you will go on the apheresis machine that will take off, in this case, your T lymphocytes. Then where it becomes different is these T lymphocytes are sent away, and right now I'm talking about leukemia lymphoma. These T lymphocytes are sent away to a centralized manufacturing factory where they are engineered. They are altered through insertion of a virus that then makes the cells recognize more strongly the specific cancer cell. So CD19 is the one that we have the most information on, is the approved product for leukemia and lymphoma. Once you've engineered the individual's own T lymphocytes, then you grow up. You grow them up, proliferate them, make an army of cancer-killing T cells. Then they're sent back to wherever the individual is. They get chemotherapy similar to your conditioning before transplant, and then the CAR T cells are reinfused. So that's the process. Now, clearly there are a lot of steps to it, and it's complicated with multiple levels, but in leukemia and lymphoma, at least some types, it works. And it's actually been astonishing what we've seen. In completely refractory acute lymphoblastic leukemia, around a 90% response rate. So that's where everything else has failed: chemotherapy, radiotherapy, transplant. These CAR T cells appear to be completely eradicating the disease, at least for a while. Lymphoma, we're not quite there yet. It's probably around 50%, although varies between studies. And definitely a lot of attention is turning to moving these treatments up from your last ditch attempt to maybe earlier in treatment, and also to trying to design new CAR Ts that may benefit other diseases, such as myeloma. Now, the limitations are still pretty big. We have a lot of work to do here. The side effects, as evidenced by Emily's case, can be really severe. Cytokine release syndrome, which is massive inflammation, means a significant number of people end up in the intensive care unit because they become so unwell. It can also cause nerve issues, transient usually. The cost of around $400,000 US per dose. Even in the US, that is causing major challenges. And that's just the living drug itself. That's not all the other parts of treatment, being in hospital, the treatment for side effects, all these other aspects. And even once it works and you get a complete response, it doesn't always last as long as we'd like. For Emily, it's been brilliant, and for some people it is. For others, maybe the tumor gets cleverer and says, you're after my CD19. I'm going to drop that and I'll get on without it, meaning your CAR T cells don't work anymore. Or maybe the CAR T cells don't continue to remain in the circulation, and that could contribute to treatment failure. And right now, access to treatment is a real challenge because it's so complex and so expensive. Now, specifically in myeloma, we are looking, not me, the royal we, looking at BCMA as a target for CAR T cell therapy. And in phase 1, there have been really promising results so far, not dissimilar to what we've seen in ALL. But these have not necessarily been sustained. And phase 2 studies are ongoing, including one being led out of Toronto, looking at the BB2121 drug. So this is something where I think we may yet see a lot of promise. There's a lot of excitement, but there's an awful lot yet to be learned and improved. Because right now, I think we're at the Model 4T stage. And I'm hoping, this is projection, that we can really improve on this. There's all sorts of things that can be done, such as inserting safety switches, so if you get a really severe side effect, you can turn it off. Maybe targeting 2 different antigens. What about if we went for BCMA and CD38 to try and really get it? But this is all into the future. This is projection and hope. I suspect we are just starting to ride the wave of immune therapy. And I'm quite excited to see where it goes. So overall, BCMA-targeted therapies, there are lots of different types in testing. Antibody drug conjugates, BiTEs, CAR T cell therapy, very promising in very early studies. And we need to watch this space and try and contribute to improvements in further learning. So overall, I think there are really, really good reasons to be hopeful in myeloma. We've already come a really long way with all the advances, even just over the last 10 years, that have made a huge difference in survival, with the average now increased from around 3 years to around 8 years or more. We know you want more than 8 years. Recalling again, that is the median with a range. So lots of efforts continue to improve further on that and possibly turn myeloma into a curative disease. And certainly that's where the discussion is going at senior meetings. How can we cure myeloma? Not how can we knock it down for longer and longer. So hopefully we have some time for questions if you have any, but thank you for your attention.
Wow. A key takeaway for me is how Dr. Hay described how these new and emerging therapies are enabling us to have longer and healthier lives. So we as myeloma patients really applaud the efforts by our great doctors, researchers like Dr. Hay. So we do have time for a few questions. Yes, Sophia.
Thank you for great talk. Regarding venetoclax, the original was as a single agent, but the one that the FDA stopped, it was in combination. Would that additional drug cause that, or they saw that pattern in the beginning also?
That's a really good question. Just for those who may not have heard it, Sophia's pointing out that the original study I showed you by Saji Kumar was as a single agent of venetoclax on its own, whereas the subsequent Bellini study was combination, and wondering if that makes any difference. Now, based on what we've seen so far, there is actually concern about all of it potentially, and I know many physicians are not routinely prescribing it in the US in any situation right now until they learn more. But there are so many unknowns, and we just don't have the answers at this point, but we're looking for them.
Thank you. Excellent talk. And I think there's, oh, sorry. The green light's on. Okay. There's lots of reason for hope, but also lots of reasons for thanks. One of the questions I had is a lot of the treatments are targeting the plasma cells themselves. Is there something upstream, like a conductor that's telling them to turn on and proliferate? And is that where we should be targeting? Maybe it's inflammation, but is there something directing the orchestra upstream?
That's a really good question. I wish I knew. So, so I think what you're getting at is, and it's one of the things we always look at, is there maybe a stem cell that has gone wrong that is then driving this downstream? Now, not as far as I know right now, but that's not to say there isn't, and that's not to say there may not be others looking at it. And perhaps if others in the audience who may be working in this area have additional answers, we can address those later. It is good to try and figure out what one cell went wrong and how can we address that and what may be driving it.
Yes, Robin. No.
Good presentation and very clear and succinct. I have a question with respect to clinical trials and the fourth phase clinical trials and sort of what I guess is becoming more and more real-world evidence. How does this really work in the world? And I'm wondering, can you give us an example of a trial that's gone that far once the drug has been introduced into the market, and whether or not patients are engaged in that and kind of looking at not just the health aspects, but the quality of life aspects that these drugs, because sometimes that's more important than just having an extra month of life, quite frankly.
I agree completely. You guys ask good questions. So the example I can give you right now at our institution would be in chronic lymphocytic leukemia, where there is a Canadian DEVOTE phase 4 clinical trial, and it is venetoclax based. So it is for individuals who are getting venetoclax as we would for standard of care, but asking the patients, because this is always consent, you are not just enrolled in a study without knowing all about it. So saying to people, do you mind if we collect at a high level some de-identified information about how it actually works for you and your side effects? And also, would you please complete some quality of life questionnaires so we can really understand how this is impacting you, either positively or negatively? So that would be an example of a phase 4 study in chronic lymphocytic leukemia. They're much simpler because you are getting a drug that you believe already works, so they doesn't have all these other additive things, but it certainly adds to knowledge, especially about quality of life.
Hello, okay. Do you have any insight into the venetoclax, the increased deaths? Was it related at all to like tumor lysis syndrome? Do you know at all what the causes were?
Another good question, because yes, in the chronic lymphocytic leukemia early studies, tumor lysis syndrome was a major problem with venetoclax and did actually unexpectedly lead to a couple of early deaths. Then it was very quickly addressed, and in any venetoclax study now, there's a very careful ramp-up phase and close monitoring so that doesn't happen again. So it doesn't seem to be explained by tumor lysis syndrome in the Bellini trial. The other things, now, I should say I do not have access to the full data. Right now we have an abstract and we have an FDA release. We do not have the full clinical trial data. But from what I understand, there wasn't a major difference in infections or pneumonias or tumor lysis. So it has us all really wondering what's going on here.
Thank you.
Yes, any other questions? Well, I have just a quick comment, again, in appreciation for the work that Dr. Hay does and our other researchers. You described research and advocacy, and those are two of the most important objectives of Myeloma Canada. And that has helped put us in Canada, as Dr. Rees mentioned yesterday, in a very, well, it could be better, but a good situation compared to most other countries in the world, including most rich countries. And it's because largely of the work done by Myeloma Canada and supported and collaborated by the great doctors, myeloma doctors like Dr. Hay, that we are in this position. So again, thank you again, great doctors like Dr. Hay, and thank you, Dr. Hay, for your great presentation of some of the emerging treatments coming out, and also a good description of how we go about research to develop those treatments.
You're welcome.